So thanks, Casey, for the intro. And it's a pleasure for me to give you a lecture about R, bioconductor, and ggplot today. So it's a suite of things that's going to boost the productivity in terms of genome data analysis and visualization, right? So first, so regarding to outlines, I will first talk about the programming R, like some basics, like R grammar, basic, like just like Casey introduced bash, I'll talk about some of the basic for R as well, like variables, data structure, control flow, all kind of stuff. And then move on to bioconductor. So it's, as Casey said, it's a package repository, mostly for bio-related packages or for high throughput data analysis. There are a lot of packages like very useful for genome data analysis under bioconductor, and I'll use one example called biostring to showcase you how to use package from bioconductor to do sequence handling, manipulation, and or things like that in live demo. All right. And the third part I'm going to talk about is ggplot. It's a package for data visualization. I would say it's kind of interesting that some people even choose R to do the program because they can use ggplot. So this is how they're so useful to do data visualization. It's just kind of like, it's hard to get rid of it once you start using this. I will talk about some of the basic ggplot, like the foundation, the theories behind it, and also the basic structure, how to code in ggplot. And then I will do some live demo, like just to show you how to do this in real time. And then if I have time, I will talk about ggtree. This is actually a package that is built upon the ggplot foundation. So it's using the same idea, the same theory, but it sort of extends ggplot's capability for phylogenetic analysis in specific. So if you are studying evolutionary genetics or phylogenetics, it will be very useful to know ggtree to help you do more advanced phylogenetic analysis visualization. All right. So start with R programming. So I tried to design this course so that people who doesn't even know anything about R should be able to understand it. So just try to follow along. It's gonna be a ton of information. So it's okay, you don't have to follow everything I said. You can just always go back to the slides later or ask me on Slack after the class, right? So to make sure everybody's on the same page, I'm gonna talk about what is R. R is a language and environment for statistical computing and graphics. So I highlight those two parts. Those are the two main reasons why you want to use R for, okay? So first, if you like, let's say you're doing statistic analysis, some basic like linear and nonlinear modeling, classical statistical tests like Fisher test, chi-square test, F-test, or you want to do time series analysis or classification, clustering, or you want to do some advanced statistical analysis like simulation, like Monte Carlo simulation, you want to use R because it has a lot of built-in functions in R, in core base R, that helps you do this kind of task very easily. So you don't have to write 2,000 lines just for neat linear modeling. So you can just probably do that in one line. It's very easy to use. Also about graphics, R is well known to produce publication quality plots, right? You probably already know this, and a lot of the paper have plots you probably know already are generated by R, right? So R has some base functions to do graphics, to produce plots, and also because you have ggplot, such a powerful package or like an extension in R that you can do some data, like advanced level data visualization. So this is kind of the reason why R is so famous for graphics like production. And so I bring out ggplot, and this is just one of the many packages you have access to once you start using R. So there are probably around 20,000 packages at least available and published for R. And each package is designed to do a specific task. And there's a suite of packages that are just designed for genomic data analysis, and mostly they're in bioconductor. So this is just, once you get to know R and start using those packages for your day-to-day analysis, you'll find your productivity is a major boost if you want to do genomics, right? And another point I want to bring out, probably not going to be the focus of my lecture today, but R is kind of becoming the center of facilitating reproducible research nowadays. It's kind of interesting. So what is reproducible research? It means that in the future, you and anyone else will be able to pick up your analysis script and try to reproduce the same results, including figures and tables, right? It also implies well-documented codes. So all your codes will be well commented, and the reason behind the functions and methods will be described in a single report. So you have a report that have codes, words, and sentences together. And basically it means that all the numbers in your report are not you're not typing that number down, because it will raise human errors, right? They're all generated by code automatically when you try to generate the whole report. So it's going to be super reproducible, and it helps your own in terms of lab research, and also it helps others when you try to publish your work. So it's a pretty good concept for genomic data analysis, and there's a package called R Markdown. So you can use this package to create a report that have all these functionalities to boost reproducible research. But unfortunately, I don't have time to cover this today. But in this link, it's kind of like there's a book for R in reproducible research. Definitely use this if you're interested to cover this section. And actually, this whole book is written in R. So how cool is that, right? And it's actually written in another package called R Bookdown. So you can write book in R. Keep that in mind. Anyways, just some basics for R, and then I want to talk about how to use R. So for people who doesn't have R right now on your laptop, you can download R from CRAN, C-R-A-N. It's a network of FTP and web servers around the world that store identical up-to-date versions of code and documentation for R. And also most R packages. There are around like 15,000 packages currently being maintained by CRAN. So the servers will be in different locations of the world. There are like probably at least 10 servers around the United States. Like there was one in Georgia, there's one in Ohio States. So yeah, just it's to make sure people can download R as fast as possible, right? So using this link, you'll be able to download R from CRAN, and you can download R from Conda. So Conda is an open source package management system and environment management system. It helps you to create a specific environment for a specific task, let's say RNA-seq analysis, and help you install all these software dependencies, so you don't have to install on your own, especially if you can't find this package in let's say Ceph 2, or they don't have this module available. You can use Conda to easily replace that. So I won't talk about this today, but Thursday Preston will talk about Conda in detail and how to use it, right? So if you have R already in your system, you can use R in command line. So in terminal, just type R. So it'll pop up R console, and then within the R console, you can just do R stuff, like type R command, just like when you type bash command in the bash terminal, all right? And most importantly, I think commonly people will use R Studio to program in R. So it's an integrated development environment, or IDE. So it includes a console. It includes a syntax highlighting editor that supports direct code execution. So you can actually write code in editor and execute it line by line. So that's going to be very helpful if you want to do debugging. And also it has tool for plotting, history, and workspace management. It's a very useful IDE for if you want to program in R. So you can use this link to download it. It's free. The basic version is free. It definitely has a commercialized version, but you can use it for free basically. Once you have R and you have R Studio, you have some script, you can run this script. So after you install R, you should have this R script command available in your environment variables, environment path in Bash. So you can just like in Bash terminal type R script and then space, and then try to add your the file name of your script after it. So then in this way you can execute R script and generates an output in the standout. And also you can just directly execute this R script. But keep in mind that I think similarly, when you try to execute a Bash script, you need to add some appropriate shebang. In this case, it's going to be the hash and exclamation mark followed by the pass to the R script, right? So if the R script is already in your environment variable, then you don't necessarily need to add this. But yeah, if you want to execute it directly, you need to add the shabam. All right. So this is how to use R. And next, I want to talk about how to, use how to like understand some basic R grammars and how to use it or do scripting. So first is mathematical operations. So in R, all the conventional mathematical symbols like plus sign, minus sign, star sign, and slash are used for addition, subtraction, multiplication, and division. So it's gonna be commonly used in many different languages. And actually in Bash, you use the same symbol, right? This is some example how to do basic mathematical operation in R. So you don't really need to add the space surrounding this mathematical symbol, but it's recommended. So it's kind of like, it's a style thing. So adding the space, it's easier to read the code, right? So in Bash, you use the same symbol. However, the grammar is going to be a little bit different. You need the $sign followed by 2 parentheses, and you put the mathematical expression inside this structure to make sure Bash can interpret this mathematical expression correctly. All right. So I just want to show you the difference here. In R, just make sure you know that space is not necessarily needed. All right. And then about comments. So again, pretty similar to Bash, R interprets anything on a line after this hash symbol as comments. So anytime you have this line start with the comments, this line will be ignored. Okay. So when the R try to execute the whole thing, this line will be ignored. And this commenting is very commonly used in programming languages. So it can be used to add text in the program that will not be executed, and they're useful for providing context to describe what the code is for, or like what this code does, right? So or even you can use it to temporarily get rid of a line when you try to debug stuff. So okay, maybe this line is problematic. I want to get rid of it right now. So you can just add a hash at the beginning of the line. All right, and you probably noticed at the final line includes this print stuff to print final results. So this is actually something I want to talk about next. It's a function in R. So the R function is created by using the keyword function. So this is a structure to create a function. It starts with function name, and you have this specific symbol. You have to care about this right now, but it's basically you have function followed by parenthesis, and you put in the argument followed by a curly bracket and put in the function body. So this is how you create a function, but you don't have to know this right now if you're not familiar. You can just use a function. Other people write, or probably it's included in base R already. So to use a function, this is probably more useful. It's a function name and then followed by a parenthesis. So in the previous example, I show you actually a print function in base R. So this is like print parenthesis, and you put the stuff you want to print within the parenthesis as arguments, right? So this is how you typically use a function. And so let's say a function exists, but you don't know how to use it. So you can easily use how function, and within the parenthesis, you put in the function name, or even easier to use a question mark followed by function name to check what kind of functionality is provided for a specific function, like which argument can you use, how do you tweak options, and also example codes for this function. You can do this in RStudio or in R console, all right? And there are many built-in functions in R that covers a lot of different stuff, like input and output, data creation, slicing and extraction data manipulation, math, matrices, string, and plotting. So it's gonna be a lot of different functions, and it's hard to get to learn every single stuff before you start programming R. But as Casey mentioned before, it's always good to sharpen your sword before you actually use it. So this link covers, it's actually kind of like a reference or cheat sheet to cover like majority of the base R functions categorized into different sections. Like this part is for math, and that another part is for matrices. So be sure to check this out to scan through this, at least those functions exist, And later, if you want to use it, you can just go back to this reference to easily pick up the function you want to use, okay? So functions. Next, I want to talk about variables. So I think Casey also introduced variables in Bash. So it's similar in concept. It's an object that stores piece of information. You can either store single value or multiple values organized in a certain data structure. So in R, you assign values to a variable using the specific operator. It's an arrow sign made with a caret and a dash. So this here I show you example of how to assign values to a variable. So so you start with the variable name. In this case, it's var, followed by the assignment operator, this arrow dash, and then the actual contents of the value. It's recommended to use this specific operator. You can also use equal sign in R. And also, by the way, you don't have to use space, but it's recommended. However, it's less preferred. Make sure to use this arrow sign in R. It's a style thing. It's also kind of like you have some style in Python you want to stick to. So it's the same thing for R, just to make sure to use this way instead of equal sign. So in Bash, you use equal to assign values to variable. And the key thing about the difference here is that in R, you can do this. However, in Bash, it's not allowed to have space surrounding the equal sign, right? If you have space, the whole thing will just fail, right? So to make sure you know the difference here. Let me see. After you assign values to the variable, you can easily just type the variable name in R console or in R Studio to show its value. So in this case, if you type var in the console, the output will be being 8940. It's a string here, right? So it's this is a basic for variables. Next, want to talk about data types. So R provides some basic data types that can be used to represent different kind of information. So there are numeric data type to represent numbers, there are character data type to represent strings, and there are logical data type to represent Boolean values. There are more data types actually, there are complex data types, but these are the most commonly used one, all right? So the numeric data type in R is a class that represent numbers that can be integers, like 1 or 2, or decimals, like 1.1 or 3.0. So here I show you an example of how to assign a number data type to a variable. So it's pretty easy, just variable and the assignment operator followed by a number, right? So it can be 3 here or 3.2, doesn't matter. And a very useful thing you can use is this class function. You can use this class function to check the data type within a variable, right? So if you do class var1, the results will be numeric because you're putting the numeric data type for this variable, right? And then characters. So character data types include all string values, and a character is denoted by its surrounding quotation marks. It can be either single or double quotation. Just to make sure people understand it, if I have a number surrounded by double quotation marks, it's going to be interpreted as numeric or character. It's going to be characters, right? Every time you have this double quotation mark surrounding something, it's going to be a character. So this example shows you how to assign a character data type to a variable. And then when you check the data types for this variable, it's going to be character, the final results. And then logical. So the R logical data type has 2 possible values, true or false. It is important to capitalize this TRUE or false, FARSE, and make sure you don't have quotation marks surrounding this, true or false. Otherwise, it's going to be interpreted as characters. So this is how you assign logical data type to a variable. And if you check the class, it's going to be logical, right? It's the class thing is very useful when you don't know what's the data type for this class. You can check this easily. So this is basic data type and multiple. Let's say you have multiple values. So elements of these data types can be combined to form data structures. This is probably going to be more useful when you move along and start using R more and more. It's just less common to use single value than just a complex data structure. So you can, first I want to talk about vectors. A vector is the most common and basic data structure in R. It's basically a list of values, mostly either numbers or characters. And you can assign a vector to a variable, just like you would do for a single value in the last slide. And here I show example of how to create a list, how to create a vector for a specific test. Say you want to create a list of species name. So basically, you can create a vector using this C function. So you start with a C, and in the parentheses, you're putting a set of characters. It can be characters or numbers, doesn't matter. Just you're putting multiple values, it becomes a vector. And you can assign this whole vector to a variable, in this case, species. And when you check the class, like the kind of data types in the species, use class species, the results will be character because you put in characters, right, as elements for this vector. You can also, just do this for numbers. So another task, let's say you want to create a list of species genome size for E. coli, human, and corn. So you can also create a vector for numbers, C, putting numbers, it can be decimals and integers, and you assign this vector to G length, this variable. And when you check the class of G length, it's gonna be numeric, right? Because you put in numeric values. So now I wanna ask a question. If you create a vector including both integer, decimals, and characters, let's say it's 4.6 and human and corn. So when you check the class of this variable, what's it gonna be? Anybody have an answer? It's gonna be characters, all right? So even if you put in numbers, everything will be, recognized as characters universally. So you only have one specific data type encoded for a vector, all right? So if you put in characters, everything will be considered as characters, all right? Also, you can do math with a whole vector. So let's say you want to double the genome size, you want to check it. I don't know why you want to do this, but you can totally do this by using 2 and like operation, this multiply operation, followed by G length, which is a vector of numbers. The results will be, as you expect, 9.2, 6,000, and 100,000, right? So once you learn more about R, you will find this vectorized concept happening everywhere, not just for vector, but also other data structure as well. And this concept will make computation much more efficient. Just keep that in mind. So it works for a vector and it works for many other stuff, all right? Next, I want to talk about data frame. It's another data structure. So even though vector is sort of the basic and foundation, data frame is probably your best friend. You gotta use data frame every single day if you want to do our data analyses. And it's gonna be very crucial for plotting as well. So it's very important to know data frame. So a data frame is an R object that stores data in 2 dimensions. So remember, vector is just 1 dimension, right? 1 dimension of numbers. But data frame is 2 dimensions, similar to matrix, I would say, represented by columns and rows. Each row of the data frame represents a unique set of observations. So that's actually a crucial point that separates data frame from matrix. In data frame, every single row is a unique set of observations. So this is actually one example. This is actually the variables that's built into R. You can check this out by just typing empty cars in your R Studio or R console. So this is data table showing you all the features for a specific car model. So each row is a car model, right? And every different column is a specific feature. So top line of the table is called the header. So include MPG, CYL, you see. So it contains the column names. And each horizontal line afterwards is a data row, which begins with the name of the row, in this case, a car model, also an index for this row, and then followed by the actual data. And each data member of the row is a cell. So in this case, 21.0, it's a cell. It's important to know like the basic concept of data frame, so you can refer to this in the future. And even though, as I said before, even though vector is more basic, data frame is going to be most commonly used data structure for your own analyses. and it's crucial for plotting. So I'll definitely talk about this in a second. So definitely check this out. Just learn data frame as much as you can. So probably it's less common to create a data frame in R and type all the numbers in RStudio. It's more common to have file already, let's say a data table in CSV or TSV file format, and you read the file into R, right, as a data frame, and then you can do some manipulation and visualization afterwards, right? So here I want to show you a basic workflow of how to read file into R as a data frame. So commonly the data should be from some other software. So it's probably like a VCF file or GFF file, doesn't matter what the format is. But I will recommend you to prepare data in comma separated values. So they're also TSV files, but the tab delimited situation, those tabs sometimes can be misplaced with space, right? So if you see those file formats, hard to sometimes hard to parse if the delimiter is misplaced. So to make sure to use comma separated values, it's commonly used in bioinformatics data analysis. And if you have the CSV file ready, you can just create a new variable. In this case, a path. It can be other names, and you assign the path to the file to this path variable, right? And then you can use this read.csv. It's an R built-in function to read the file as a data frame, right? So in this case, you use read.csv. In the argument, you put in file equal path, or you can directly put the path here. And then let's say like that's kind of the single required argument for read.csv. There's some optional arguments, some most commonly used one. If you don't have headers in your CSV file, you can put headers equal false. Otherwise, the first row will be automatically read as headers, right? And then if your data is not comma separated, it's possible it's got like a space separated or slash separated, doesn't matter. You can set the delimiter in this SCP option. You can change the comma to anything else, right? But basically, this is how you read file into data frame. You can redirect the output of read.csv and assign it to a data variable. And now the data should consist of all the information for the data frame. And so if you have data frame already in your environment, you can check or inspect the data frames using two functions. This head function will show first 6 rows of the data frame. All right, so it's going to be easy to check like what kind of information is in the data frame by using head function, and also you can use str function to display the internal structure of the data frame. Definitely try this out if you want, but it will show you what kind of columns are in this data frame, and also what kind of data type is it for each column. So some column may be for maybe numeric, and some column may be character. This is going to be important moving along for complex data analyses. All right. So, okay, now you have data frame. You know how to read it. You know how to keep this in your environment. And next, you want to manipulate it, right? To do some basic, filtering or summarizing or some mutation, like to get the sum of 2 columns, something like that. You can use this dplyr package or dplyr package that provides functions that perform data manipulation operations to explore and manipulate data set. So I put a link here. You can check the dplyr website if you want. It includes all the manuals and all the description for the functionality of this package. But basically, dplyr is a grammar of data manipulation, providing a consistent set of verbs or functions that includes mutate, to add new variables that are functions of existing variables, or select if you want to pick variables based on their names, or filter to pick cases based on their values. Let's say you have a column of people's height. You only want to select if people's height is over like 10 inches, something like that, or summarize to reduce multiple values down to a single summary. So this is kind of be a little bit complicated. Definitely you want to learn more about this or try this out to actually know what those function does and arrange to change the order of the rows. So there are a lot of different functions for data manipulation. And if you want to know more about how to use this, instead of reading manuals, which is going to be a little bit tedious, I highly recommend this chapter of the book. So this is a book written by the chief scientist from our studio, Harley Wickman. And this book is called R for Data Science. It includes all the basic stuff of how to prepare data set, reading to R, do data manipulation and visualization. And there's a chapter called Transforming Data. So it includes everything about this deep learning package, about how to use it in real scenarios. So definitely check this out and try to read it. It's also written in R, and it comes with all the code base and examples. You can actually copy and paste it into R to try yourself. So definitely try this out if you want. But I won't talk about it in detail today. Then moving on to talk about control flows, I want to talk about how to do for loop and conditional statement in R. I think Casey already mentioned how to do this is in Bash, but it's gonna be similar concept, but the grammars is gonna be a little bit different. So I show here how to do for loop, conditional statements, and combine them together in R and in Bash. So in R, let's say you want to do a for loop to, in this case, you have a G length, which is a vector of species genome size, and you want to loop through this variable to print the genome size individually, all right? So the for loop statement goes inside the parenthesis after for this FOR, and it's followed by a set of curly brace that contains the code to be executed. So this is the basic structure of for loop in R. And it's the same thing for conditional statements. In this case, I have a number, and I want to check whether number is greater than 10. If that's the case, execute this print function to print n is greater than 10. Again, the conditional statement goes inside the parenthesis, followed by brackets. And within the brackets, you do the execution line, all right? So both for loop and conditional statement, the structure is very similar. And you can combine those two into one nested structure. So in this case, I want to check for every single element in this species genome size vector, is it greater than 10? If that's the case, print out this element. So you can start with a for loop structure. Within the brackets, you put the conditional statements, and so you have two set of brackets here. So it can be easily combined, and it's going to be very easy to use in R. In Bash, the major structure is similar, but the grammar is a little bit different. I just want to put it here so you understand what's different about doing for loop and conditional statements in Bash. So Casey already mentioned this. I won't go through this again, but you can go back to this slide in the future to see, okay, like something is different, all right? So after control flow, I'm going to talk about packages. So as you already know, one of the reasons you want to use R is because it has so many useful packages available for different domains, and there are a lot of packages for genomic data analyses, right? So R packages are bundles of code, data, documentation, and tests that are easy to share with others. You have base packages automatically installed when you install R. So I talked about this before. There's some built-in functions. Some of those functions are bundled in a specific package. And CRAN, as you know, it hosts most common R packages available. And to install packages from CRAN, you can use this install.packages function. So basically in console, you just type install.packages. Within the parentheses, you put in the package name, if you already know this. So basically, majority of the package will be available in CRAN, and you should install packages in this way. And also, bioconductor has a set of packages for the analysis of high throughput genomic data. So if you want to do genome analysis, check the packages, check what kinds of packages available in bioconductor. The installation is going to be different. I will talk about it in a second. And also, there are some packages available in GitHub. It's organized as repositories. The installation is a bit different. You use DevTools package to install packages from GitHub repositories. It's going to be you probably won't use it very much, but I just want to let you know that some packages are available in GitHub. All right. So basically, most packages available in CRAN, most bioinformatics packages available in bioconductor, and some in developed or just kind of like a not published packages are available in GitHub. All right. So right now you probably don't know a lot of packages. So to discover or learn the right package, first of all, you can learn R, right? So there are books for R, and in those books, they mention a lot of packages useful for different scenarios. So you can learn R using this book again, like I mentioned, R for data science. So previously I mentioned a chapter about data transformation, but you can also read the whole book, scan through the whole book to see, okay, generally, when I want to do this, what kind of package is available. Also, you want to Google. You can Google what you want to do and add R as keyword. So let's say you want to plot a heat map from RNA-seq data. You can just do R, heat map, and RNA-seq. You're going to see a lot of posts from maybe Stack Overflow or BioStar, or just someone write a blog post about a specific package. So those are going to be very useful, and definitely use this method, which is going to be universally applied. Once you have the package installed, you can load the package using the library function. So you put the package name inside the parenthesis. You don't have to add the double quotation sign for this package name. Just keep that in mind. And also, after you load the package, you can check the functionality provided in the package, again, using the help function. So previously I mentioned you can use a help function to check how to use a function. You can also use help function to check what kind of function are available within a package. So you use this package argument. So package equal package name. So for example, if you want to check the functionality for ggplot, you can do package equal ggplot here, all right? So this is all I want to say about base R. If you have questions, you can ask now. And if not, I will just move on to talk about bioconductor, all right?
Benoit, can I jump in for a second? I want to just say a couple things. Excellent, thank you very much. Question about how to, there's a question about, yeah, Daniel replied already. Sabrina asked, do you have to reload the packages? So first of all, just be aware, when you log into the teaching cluster, R is not available in your base path. You need to module load either a specific version of R, okay? There's lots of different flavors, 3.1, 3.2, 3.6. So there's 3.6 and 4.0 on the teaching cluster. Stick with whichever version you use, because in fact, the packages that you load are specific to that version of R, all right? So just be aware, all right? So yes, you'd need to go onto the teaching cluster, load R, and then inside R, when you do that, it will change the prompt and it will be another command line interpreter. It's another interpreted language. The prompt will change. And at that point, you would need to load the package. First, you need to install the package, then you need to load the package, okay? So you need to install it one time, but each time you run a script, you would need to load it. So in practice, what I'd recommend when you want to do an R script as part of a pipeline, to actually have your R, you would have a bash script and you would separately have an R script, and you call the bash script. So you call the R script from the bash script. And in your bash script, you would have the module load command, and then you would have the R script R file, and then it would execute in R those commands. Okay, does that make sense? So you need to recognize that what Sean was talking about is in the context of already having loaded R. All right.
So is that like separate then? Obviously for like RStudio, you can do it on your laptop or your computer, but for this, is it like if you had big giant data sets that you couldn't feasibly do that way, you need the teaching cluster? Okay.
Correct. So, this is going to be up to you. Your development probably still would be in RStudio. And sometimes you'd have data files that are processed sufficiently that you can pull them down onto your laptop and run the R code in RStudio. But even then, you're running it manually. So there's chances that you'll get it wrong or you don't remember what you do. In my opinion, it's always better to have the last step of your pipeline be a command in your bash script that loads R and then runs your R script, and then all your files are produced automatically as well from that pipeline run. Okay. So that's just one thing. Another, you can build a whole pipeline in R. I'm not saying you should do this. R can also allow you to run Unix commands using what's called a system call. All right. So you can kind of use an R script to grab something in Unix and do a Unix manipulation as well. All right. But just be aware that that's a possibility. There's a way to kind of go back and forth between the two systems. So yeah, so I think that's one more thing. Shenhua, maybe you could, you're going to show when you do some of your interactive stuff, if the index comes up, like when you report, you print statement or you show what's in a variable name in the index, clarify that, because that's actually super confusing.
Okay, gotcha.
All right. Yeah. Yep, that's it. Okay, great. Thanks.
All right.
I'll shut up now.
All right. So for people who already know R and use R commonly in your data, in your workflow, the previous section is probably going to be a little bit boring for you. So let's move on to the real stuff. All right. So for bioinformatics data analysis. Okay. So bioconductor. So bioconductor, as I mentioned before, it provides tools for the analysis and comprehensions of high throughput genomic data. So there are a lot of packages available. I think there are 2,000 packages currently available in bioconductor. I checked it yesterday. The functional side of bioconductor packages includes, DNA array, microarray, sequence data, SNP data, and all the others. So it's going to be a broad areas. So, there's some pretty famous packages you probably already know, like BioString for handling sequence data, reading FASTA, FASTQ, try to manipulate it, or DESeq2. This is probably more well known. It's for differential expression RNA-seq data analysis, and you can use this link to check out what kind of packages are available. This is going to be very useful because all the packages, they're categorized in different sections. So, for RNA-seq analyses, for microarray analyses, for comparative genomics. So you can target your package easily under this link for specific questions. So, okay. I will not cover everything about bioconductor packages. So I will just choose one package as an example to showcase you how to, like unleash the power of some specific package for a specific task for genome analyses. So this biostrings, this package contains classes and functions for fast manipulation of large biological sequence or sets of sequences. So there are a lot of functionality available. I'll just give you some example. You can use this lens function to count the number of letters in the sequence. You can use subseq to do a subset sequence based on coordinates. So let's say you want to only select first 10 base pair of all the FASTA sequence you have, or you have a set of sequences, you want to get the complement or reverse complement, or you want to translate the nucleotide sequence to protein sequence. There are a lot of functions available for that, for sequence manipulation and transformation. And then, yeah, just saying this is probably a little bit useless. I will just show you in real time, like in RStudio, how to use this, all right? Let me switch to the RStudio console. Can you guys see the screen in RStudio? Yeah, okay, right. So key things before you try to use package or install packages from bioconductor, you need these two weird lines. It's super weird and you probably don't understand right now, like this first two lines. You don't need to understand it, okay? There's a set of reasons why you need to install packages in this way. It's weird. I won't explain today, but if you want to know the detail, you can check the bioconductor website. They have description for that. Anyways, yeah, you can copy and paste this code from bioconductor website to your RStudio, so you can just execute it easily. So it's weird. I can't use my mouse. Let me try again. All right. Okay. So basically, just execute this, and then you should be able to install packages. So basically, this is a grammar to do that. You start with bioc manager, followed by 2 colon, and then install. And under the function arguments, you put in the package name. So in this case, biostrings. Because I already have biostring in my environment, so I won't execute it right now in my environment. But after you have it installed, you can load the package into a current environment by using library function. So library biostring, let me try to execute it. Oh, by the way, so for executing a single line in RStudio, you can either use this run this, just after you select the line, you can use run to just run this part, or just the multiple lines if you select it, or you can just easily just type command plus return. This is in MacBook, and in Windows, it's probably control plus return, if I'm correct, right? So in this case, I would just type command plus return. So it will execute library biostring, and now it's in your environment. So one of the functionality for biostring is to read the sequence and do some transformation. To read the sequence, you can use this function called read DNA string set. It's a little bit long name, and you can check out as I mentioned before, you can check out the function usage by doing help function and putting the function name, or easily just type question mark followed by a function name and hit enter. And you can see RStudio will just pop up the help page for this specific function. You can see that this is a function to read FASTA or FASTQ files in a specific object. You don't have to know everything about this object, but it's organized in this specific data structure. So let's try to execute this. Basically, you put the read DNA string set and use the path for your FASTA file. You put it in and you execute it, and you redirect the output into to assign this to a variable, which is DNA in this case. So now we can check the content of this DNA variable just by typing DNA, right, and hit enter. So you can see this is how RStudio displays this specific data object. It includes width for the sequence, the actual sequence letters. It's actually highlighted in different colors for different nucleotide, which is pretty cool. And also the name of the sequence, which is actually the FASTA header. If you have multiple sequence in a single FASTA, it will be displayed into different rows, right? So this is how you read a sequence. And then let's say you want to do some sequence transformation, like want to get the complement of this DNA sequence, you can use the complement function. Again, just putting the object name, in this case DNA, and assign the output to another variable, in this case DNA complement. So just execute this line and then check the DNA complement. You can see everything is same, but the sequence is actually the complement of the original sequence, right? So it's pretty cool, and it's pretty fast. So you can do complement and reverse complement. So there are a suite of different functions available for biostrings. You can definitely check this out, like using, as I said before, help and package equal biostrings, right? So in this help page, you have the description, you have user guides, and all the functions available for this package. You can see there are like tons of different functions, right? You can, and nicely, I think some packages, they have user guides written in different documents. So like, if you want to check out like the quick overview of biostrings, you can just easily click that, and you'll see a PDF file, which is kind of like a small document describing, how to quickly use some of the function for different tasks. All right, so it's going to be very useful. Finally, after you've done the sequence transformation, let's say I finish this reverse complement sequence and assign it to this DNA reverse complement variable, and I want to write this variable out to a new FASTA file, right? You can use this write x string set function to write new sequence as a FASTA or FASTQ. So you put in the variable name, in this case, DNA reverse complement, and also the path you want to write to, right? So in this case, it's a new file path under, in my case, under downloads. You can name it whatever you want. It can be test1 or test2, doesn't matter. And you execute it, and now you should be able to see, if I go to downloads, you should be able to see, sorry, is it actually, so let me try again. You should be able to see this test reverse complement FASTA right under this folder, and you can go, sorry, the mouse is kind of getting weird. You can actually display this content using more, in case you teach that, right? So you see it's a header followed by sequence, which is a reverse complement of the original sequence. So this is a basic workflow of using biostring to do sequence transformation. And actually, as you can see in the documents, the biostring is super powerful. It can do multiple sequence alignments. It can do pairwise sequence alignments. So, if you have more complicated task, you can do this in biostring. Definitely check out their document on how to do this. Okay, let me go back to the.
Shenhua, can you just show the index there, which is something that is confusing for people new to R?
So like the index for data frame, right?
Yeah, just that ignore it, but just be aware, try to explain what it is.
So I think I mentioned what kind of data. The data frame is kind of like a data table. So R have some like data frame built in, so you can definitely just directly type the name of the variable. It's already in the environment. In this case, empty cars. If I try to type the head, like use head function, just display the first 6 rows, you can see this is what the data frame looks like, right? And this is an index basically. So master RX4 or master RX4 WAG. It's not a column. So this is before you have the columns, right? So this actually is sort of a unique identifier for each row for specific observation. So you can actually use index to target one single row easily, right? So this is, basically the most important thing is different from the columns, right? It's not in the original data frame, but you can definitely.
Sorry. I meant like in your biostring result, you have a number 1. If you go yeah. Sorry, I didn't. Yeah. So see the number 1 in brackets there. Yeah.
Oh yeah.
That just be aware is not a part of the result. It's telling you it's the first result. So like when he showed the Unix terminal, that was not a part of the output.
Yes, sorry. Yeah, I didn't.
Yeah, sorry about that. So just that's a little confusing to people. So if you have a lot of results that come to the screen that wraps around on multiple lines, you'll see another number that tells you it's like the 47th element of that list. It's not a part of your data. It's just a pointer to tell you like what row number or what line number you're on. It's the element index. So just be careful about that. That's something that people new to R don't understand. But after a while, you tend to even ignore it, right?
Yes.
Yeah.
Naturally I just ignore it. Doesn't matter to me. Yeah. So this is a live demo for biostring. No worries. I will give more live demos for other stuff later. So yeah, it's going to be more interesting coming along. So this is pretty much what I want to say about bioconductor. You can, because everybody will use different packages for their own analyses, because everybody's analyses are very different. So you probably won't use RStudio in the future, but you will use some other packages. So, but the basic concept of getting to know a package, reading the function description, and try to use their function to realize some of the tasks is pretty similar. So, that's just, you can try to replicate this for other packages as well. Next, want to talk about another package, like briefly talk about another package called ggBio. It's sort of a programmable genome browser environment. So for people already familiar with IGV or integrated genome browser, so it's basically to try to do ggplot in R. So you can code the ggplot, right? So this is pretty cool. So you can show the read alignment, the read coverage, the variant annotation, all those stuff in the integrated panel for a specific region of the genome. So pretty much what you can do in genome browsers, right? And also it has a lot of other functions like plotting some circular plot. So circular plot is commonly used when you try to visualize, structure variant, complex structure variant around the genome. For example, in this case, you have 2 different chromosomes, and those 2 lines connect 2 different regions in 2 different chromosomes, indicating a translocation event. So yeah, it just there's some of the specific genome type plotting that can be you can use from ggBio. I won't talk about this in detail. Frankly, I didn't use it a lot. I use IGV still mostly, but you can use this link to check like how to use ggBio in your workflow. And also another point. If you want to plot circular plot only, so if you want to just focus on circular plot for your structure variant, definitely try this R package called Circlize, CIRCLIZE. It produced fantastic circular plot, like very beautiful. So definitely check this out. So next, the main course, ggplot, all right? So I probably spend around at least 50% of my time are just to code ggplot script, because majority of my time is just do data visualizations if I want to use R, because other time I just do software engineering in Python. So in R, like majority of the time is just devoted to ggplot. It's such a powerful package to do day-to-day genome analysis, data analysis and visualization. So to give you some motivation for people who doesn't use ggplot right now, so this is how you plot a scatter plot using base R function. So as I mentioned before, R has built-in function to do plotting, right? So you don't have to know everything about the code I put in here, but basically this is a set of code to plot the association between two variables under empty cars that I just introduced, like a built-in data frame. You can see there are like 17 lines, and then just point for stop for a moment, and here this is a scatter plot using ggplot to do the same plotting. You can see 17 lines becomes 3 lines. It can be even if I want to put in 1 line, I totally can, right? So it's just a much easier way to do plotting. You don't have to write 200 lines just to plot a figure 1 in your manuscript. You can probably replace it with 20 lines, which is readable. And the style is actually, the default style is actually pretty good already, right? And you can do some more customization above that. And actually, ggplot doesn't only do scatter plot. It do all kinds of plot. If you want to plot scatter plot, histogram, line plot, heat map, pie chart, box plot, you can ggplot can do this, right? Probably I would say at least 90 or 95% of your visualization task can be realized in ggplot. All right, so definitely if you don't know this already, try to get to know ggplot once you dig deep into R, and it's gonna help your it's gonna boost your productivity in terms of explore your data set, right? In general, I would say. So just basically to summarize the advantage of ggplot, it's easy to use. The code is super readable, just like I think a Python code is just super readable. It's a readable language. You can change the layout easily. Just say if you want to plot a box plot, you did that, and you want to switch later to your histogram, you can totally do that with just a change of 1 line. So pretty easy to do. But you can't really change layout easily in base R. That's what I would say. It's highly customizable. You can tweak titles, axis labels, legends, all the other stuff, like even every single data point, the color of the data point, easily in ggplot. And also about scientific quality, it's just very easy to produce ggplot in a scientific standard. So a lot of paper you read, probably the plot is generated by ggplot. They have a specific color scheme. I will talk about it in a second, but you can easily, tweak a plot to make it publishable. That's what I'm saying. So that's why ggplot. And next, we'll talk about some background. So just as the grammar of language helps us construct meaningful sentence about words. So like we have grammar for R, we have grammar for Python, we have grammar for Bash, those programming languages. And there's also grammar for graphics that helps us construct graphical figures out of different visual elements. So there's this guy called Leland Wilkinson. He wrote a book and actually developed a concept called grammar graphics. The book is called Grammar Graphics, if you're interested. It's a pretty theory-based book. And another guy later called Harley Wickman, he's in our studio right now. He adapted this grammar graphics into a layered grammar graphics, which is a foundation of ggplot. So basically, this grammar graphics try to organize a plot into different components, including the data being plotted, the geometric objects that appear in the plot, like circles or lines or box plot, and a set of mappings from variables in the data to the aesthetic, which is basically saying what kind of variables do you want to map from data frame to the plot. And some other stuff, some high level advanced stuff like statistical transformation to calculate the data values using the plot if you wanted just to plot the mean value or something like that, or scale, all the coordinate system and the facades to group data showing different plots. So if you want to plot one subplot for male and another subplot for female, you can totally do that. So these are pre-structured components of plot. With all those components, you can plot any different plot if you like, right? Just by properly combine those different components together. And basically the developer of ggplot, what he does is he transfers this component of plot into different layers. So it's kind of like a Photoshop. So when you try to plot something, you start with the data, which is the first layer. And based on this layer, you just add new layers. So let's say a second layer is what kind of plot it is. Is it a circle plot or line plot? And a third layer defines the coordinate system. And a fourth layer defines something else, like the coloring or some position adjustment or scaling. So this is just an easy and readable and friendly way to plot something, and it's intuitive, right? So you want to add some stuff just like how you add stuff in PowerPoint or Photoshop. You add a new layers, build upon them, right? So this is the basic theories behind ggplot2. And so if you want to use it, if you don't have it already in our system, you can install this by doing install.packages and putting ggplot2. So this package is available in CRAN. So this is how you install the packages, all right? And then I want to talk about the basic template. So this is a basic structure, like the very basic structure. You absolutely need those core elements when you try to plot every single ggplot. So it starts with ggplot function. Within the parentheses, there are two main arguments. So you have the data arguments, which is what the data you want to plot, which is typically a data frame, all right? You also have the mapping aesthetic. This is a little bit complicated. The grammar is a little bit weird. It's mapping equal AES function, and within the function, you put the mapping aesthetic, which is basically the variables you want to plot. So let's say you have a data frame with two columns, X1 and X2. You want to plot X1 as X-axis and X2 as Y-axis. So in the mapping aesthetic, you can put X1 and then comma X2. So basically the variables you want to plot. And then you use this plus sign to add new layers to ggplot, right? Just as I mentioned before, the layered system uses plus sign to achieve that. And then it's followed by genome function. So this genome function is basically this geometric object I mentioned before, which is a component of plot. So is it a circle plot or a line plot, a box plot? So there's a specific genome function for different plot style, all right? So this is a basic template, and I want to show you the examples of how to use it. Let's see. All right. So in ggplot, yeah, you probably don't want to use this. Yeah, I just want to pinpoint that some of the packages I mentioned before, like data transformation using dplyr or ggplot2, they're all currently incorporated in this package suite called tidyverse. So this is a pretty powerful suite that includes a lot of very basic set of functions, all the way from reading data to data transformation to data visualization. So it includes all the functions for entire workflow of data analyses. So definitely, if you want to use it, you can just install packages, tidyverse. So it installs ggplot2, dplyr, and all the other packages for you automatically. And you can just load tidyverse package, or you can just you can do library ggplot2, right? It's gonna be the same. I just typically use tidyverse because I also use other functions, so it's gonna be easier for me. So if you execute this, now ggplot2 is in your environment. So in this example, I'm showing the data set that studies the species repartition and weight of animals caught in plots in the study area. So the file is putting this CSV file, and you can use read.csv or in a tidyverse way, read_CSV. It's pretty much the same, and you redirect the output into this variable called service_complete. So let me try that. And we can use, again, like I said, use head function to check the first 6 rows of this data frame. So it includes, so each row is a record for a specific, month, day, and year, and kind of the information for specific species, like for a specific object. It's a male, what's the length, what's the weight, what's the genus for a specific animal, all right? So what you want to do is, let's say you want to plot something like let's say a scatter plot. You want to know the association between the weight and the hintful length, all right? So let's build the plot iteratively. So like I mentioned before, ggplot is a layered system. We can build the plot layer by layer. First, we do this ggplot, and you put in the data set. So ggplot, under the parenthesis, you put in data equals service complete. That's the data frame you want to plot on. We'll try to execute it. So you see the plot is empty, but you have this great canvas being initialized. So basically you put in the data, but it didn't tell ggplot what kind of variables you want to put in. What kind of variables do you want to plot, right? Which is the axis, which is y-axis. So we tell ggplot this by adding this mapping arguments. So we add mapping equal aesthetic, x-axis equal weight, and y-axis equal hintful length, right? We want to know the association between those two. We try to execute this. See, now you have the weight in x-axis and length in y-axis, but you have nothing inside. So basically, currently the coordinates has been initialized. So ggplot knows what kind of stuff you want to plot, but it doesn't know what kind of plot do you want. Do you want a scatter plot, or do you want a line plot, or anything else? So we tell this to ggplot by adding this genome function, all right? So you use this plus sign to add a new layer, and then followed by genome point. So this genome point is used to plot scatter plot, all right? So this is also a function, so it counts with a parenthesis. So executing this, now we have the plot we want, okay? So the data points is now mapped from the data frame to the plot, all right? So this is a concept, building plot iteratively, and you can do some customization. Let's say you want to colorize the dots by species. This is commonly used if you have different categories of data in your data frame. So you can add some new stuff like options or option arguments for genome points. You can add some alpha values, like make each dot transparent. So make sure like when the dots are overlapping each other, you can see some information. And also adding some mapping aesthetic. So you want to colorize by species ID. So in the mapping aesthetic, you put the color equals species ID. So in this way, ggplot maps the species ID column into different colors, right? It's a mapping procedure. So it's you need to use mapping aesthetic to achieve that. So in this case, once I execute this, you can see that it probably take a second to plot right now. So each species are being categorized and colorized in different colors, and you can see some transparent dot, which is defined by this alpha value, right? So modifying ggplot is very easy, and you can easily colorize by a specific group or a specific column. Very compared to base R, you probably will spend like a whole afternoon try to do that, right? So this is kind of power ggplot. Let me try to switch back to the slides. And so basically genome function, I previously mentioned you can use genome points for scatter plot, and there's some other functions like genome box plot to plot box plot, and genome histogram to plot histograms. There are much more. So you can check all the available genome functions in this website. So this is actually the main website for ggplot2. And you can see in the layers section, you can see all the functions you can use for different kind of plot. You know, you can plot density plot, dot plot, hexagon, or jitter plot, or even maps, right? So there are a lot of cool stuff, and you can check this out in this website. And if you click on each one of them, you can see like how to basically use this, what the argument, and what the best thing is. It comes with examples, so you see what the code is and the output for the code, so you can learn this interactively. So yeah, genome functions. And next one, talk about facading, because it's also pretty commonly used. So let's say in that example, I have like dots for different species, right? It's kind of complicated to put them all in one plot. I want to have different subplot, and each subplot only contain one species. You can use facades. That's a way of grouping data plot into multiple different pieces or subplots. So there are two main functions you can use. There's facade wrap that arrange one-dimensional sequence of panels to allow them to clearly fit on one page. Or you can use facade grid that allows you to form a matrix of rows and columns of panels. So it's kind of difficult to understand by seeing the definition, but I can show you some examples how to do that. So in this case, let's say we already have this plot. Let's go back to the one that doesn't have the colorization. It's easier. So it's a scatter plot between weight and hingeful length, right? So what we want to do is we want to have two separate plots, one for male and another for female. What we can do is we can use just use plus sign. Sorry, mouse is gone. Use plus sign to add a new layer, and use facade wrap function and add facades equal vars sex. Sex is actually a column in the data frame. It has male or female, right? So, and actually, if I understand correctly, you probably can use this. So another way, basically, let me copy and paste this. An easier way is to probably do facade wrap function, and then just this tilde sign dot sex, if that works. Sorry, just tilde dot tilde sex. Let's try that. So now you have two subplot, right? One for female and one for male. They're arranged in one dimension structure. So if you have more category in this column, you will just go on and on for this row, right? You have more plots on the left side. So this is basically how you use facade wrap. So you either use facades option and use vars and putting the name of the column, or you just directly use tilde and putting followed by the name of the column. And also you can use facade grid. So let's say you want to create a subplot, and one subplot is a specific sex and a specific genus, all right? So it's kind of like you have two dimensions, right? The row is, each row is a sex and each column is a genus. So we can do this using facade grid. You can use this way. That's the default way, or an easier way is to just do sex tilde genus, all right? So let's try to execute this. See, now each subplot is a specific genus for a specific sex, all right? The sex is organized in rows, and the genus organized in columns. You can try to transpose this whole this big plot by, you know, if you want to have genus in rows and sex in columns, you can do that easily. Just try to switch their orders, and the plot will just be transposed easily, right? So you can do a lot of customization, and this facade function is going to be useful if you want to partition plots into subplots. So next is some customization. So as I mentioned before, you can add axis labels, you can add titles, you can save custom things for future use, and you can combine plots together. So I will show you some examples in RStudio how to do this kind of stuff to make sure your plot is mature and it's like readable by other people, right? So as I showed before, this is a section of the ggplot to plot the basic scatter plot between weight and hingeful length. We already add a new layer, which is a facade grid layer to partition by sex and genus. And so we want to show this plot to, let's say your PI or your colleagues. Yeah, you can add titles or axis labels to make sure people can read the plot easily. And you can also add themes to make sure the axis label is big enough so people sometimes like the default can be pretty small, so you can just make the size bigger and use some custom things. Let's try this one by one, okay? So basically this labs function can help you define the titles, x-axis label and y-axis label. Let's try that. Just select this part and try to hit run or command return. So so this plot is a plot we showed before, but this time we add the axis label and the titles, right? It's pretty easy to modify. And now, because the axis label is a little bit small probably, so we want to make this bigger. Let's try adding this thing function to modify the size of the text. So this is gonna be universally applied to every single text. So you can see this animal weight and the animal hingeful length is getting bigger, right? It's gonna be very important if you want to show your plot during a presentation. And then ggplot has this very nice thing packaged for you. So let's say you don't like to see those background lines or those coordinate lines in this plot. You can use another theme actually. Though this theme, black and white, basically simplify the plot and make sure the plot is like clean and minimalized. So if we try this, add this layer, you can see that now the coordinate system, the background color is gone. Usually it's gray, you can just delete that, and you can see that coordinate line is getting minimized, all right? So there are a lot of stuff you can do to customize ggplot, but like after you generate the data points, scatter points plot, there are a lot of other layers you can do to customize layer by layer, all right? So this is a basic theory behind it. Oh, by the way, combined plots, I didn't mention that, sorry. So let's say you have two different plots. You have this weight box plot, which is basically plotting the genus distribution. Sorry, the weight distribution for every single genus. You can do this in this section. You don't have to just pay attention to the details of the script, but basically this plot looks like this. It's for every genus, the distribution of the weight. And you have another plot actually, which is a count plot. So basically it's showing you for every single year, how many genus did you observe, right? So it will change from year to year. So that's two very separate, very different plots. Although using the same data set, but the plotting system is very different. The layer is very different. You want to try to show those two plots together in a single figure. Let's say maybe figure 1A and figure 1B in your manuscript. It's hard to do this automatically in ggplot, but you can use this package called Grid Extra. Let me try to show you. So after you install this, you can just load this in your environment. And what you can do is use grid arrange and put the plot variable, in this case, weight box plot and count plot in as argument. And you can define how many rows do you want and the width. So in this case, I have 2 rows, and for the first plot, I'll have width 4, and the second plot I will have width 6. You can customize it in very detail. So let's try to run this. See, so basically you have 2 plots arranged in a single row, and then the size of the width are predefined. So it's 4 for this plot 1 and 6 for this plot 2. You can organize whatever you want, and you can do like n row equal 1, so that there are only 1 plot per row. So like, yeah, it's, oh sorry, this is probably wrong. Yeah, but I guess I have different understanding for this specific argument, but you can definitely check like what the meaning of this argument by doing question mark, grid arrange, and you can see n row means argument of G table. So it has a specific way to organize those plots into a table-like format. So it's probably, yeah, it's probably a little bit different. But anyways, yeah, you can definitely check this out later in detail if you want. It's a very useful package to arrange multiple plots into a single plot, right? And you can also try to save this ggplot by using the ggsave function. So the main argument, the required argument is gonna be the file path for this plot and also the plot variable, right? So you have, so let's say I have this combined plot, which is the output for the grid arrange function, which is basically the two plots, right? And then I want to save those two plots, save this like arranged plot into a single PNG file. So you can define the width, the height, and even the DPI, which is basically the resolution for this plot easily, or you don't have to put in any of that. You can just do, you know, the file path and the plot variables. Just hit enter, then you can see the plot has been saved in a specific size. All right. So this is how you do basic ggplot, and I think there definitely more to it. So I will give you some additional learning resources for ggplot too. So there are documentations, as I showed you before, the ggplot main website. There are cheat sheet being produced by our studio. So you can see this is a very useful reference page. If you want to plot a specific plot style, you can just go to a section and you know, okay, which function name should I use to plot a specific line plot or scatter plot. And also, I highly recommend to you to read the data visualization portion for the book I recommended before, R for Data Science. So in this section, you can find everything about data visualization, including aesthetic mapping, facading, you know, genomic objects. So what kind of example you have, statistical transformation, coordinate system. So everything about data visualization will be covered in this plot, in this book, and comes with example codes. So very useful. So yeah, that's about ggplot. And because I feel like I already go over time. So if people want to stick around, I can show you some other stuff like some extensions like heat map or ggtree. But I guess I can also skip through because you can read you can check out yourself, you know, just by doing question mark and then function name or question mark package name to check how to do, you know, heat map or how to do how to plot phylogenetic trees. So as I mentioned before, ggtree is very commonly used to visualize phylogenetic trees, and it's actually included in bioconductor. So yeah, it's very useful, and you can see it can produce a very complex tree, well annotated with a lot of information, highly customized easily. So we commonly used it in our lab, and we highly recommend it to try this out if you want. And that's pretty much it. Yeah, this is my email address. And if you have more questions for any of the stuff I mentioned today, definitely just ask me on Slack or shoot me a question over email. We can actually all even talk about this over Zoom later. So if you want, yeah, that's pretty much it.